Good afternoon.
Good afternoon.
Please tell us your full name, spell your first and last name.
My name's Corey Crumbly, C-O-R-E-Y, C-R-U-M-B-L-E-Y.
Your witness state.
Where do you work, ma'am?
I work in the biology section at the Florida Department of Law Enforcement.
And what do you do for the Florida Department of Law Enforcement?
I examine items of evidence for potential sources of DNA, and once identified, I attempt to perform DNA testing on those samples.
Can you tell the jury about your background in education, training, and experience?
I have a Bachelor's of Science degree in molecular biology and microbiology from the University of Central Florida. I also completed a year-long training program at the Florida Department of Law Enforcement that included extensive reading, laboratory exams, written exams, oral exams, supervised casework.
And is there any continuing testing that goes on at the Florida Department of Law Enforcement?
I attend external training and read journal articles. I also take proficiency tests.
And can you describe these proficiency tests?
A proficiency test is a test given to me from an external company. I work it just like I work a regular case. I submit the results to them, and they let me know if I pass or fail.
And, strike that. Does the lab that you work at, Florida Department of Law Enforcement, is that in Orlando?
Yes, it is.
Does that lab hold any certifications?
We are accredited by ASCLD Lab, the American Society of Crime Laboratory Directors Laboratory Accreditation Board.
And what does it take generally to be certified by that standard?
We go through a rigorous inspection process where the people from the ASCLD Lab, their representatives, come into the laboratory and they look at our entire operation top to bottom to make sure that we are following international standards for quality with regards to all of the testing that we do.
And take the jury generally through the process of, say I'm a law enforcement officer or an agency, I submit a piece of evidence to the Florida Department of Law Enforcement to get tested for DNA. What process does that package go through to get to somebody like you?
The evidence is submitted in the evidence intake section of the laboratory. It's assigned a case number and given a barcode label. Then it's locked up in a secure vault until I request it for testing. Once I request it for testing, it's scanned to me through that barcode label. It's kept track of in the computer. I take it to the biology section upstairs. I lock it up in my personal locked storage while I conduct the testing. Once the testing is completed, the evidence is transferred through that barcode label in the computer back down to the vault where it is secured until the agency comes to pick it up.
And what sort of steps do you take during your examination process to make sure you are not contaminating it with somebody else's or something else's DNA?
I wear personal protective equipment, lab coat, mask, and gloves. I examine only one item of evidence at a time, and then between those examinations, I bleach down or clean the area before I examine the next item. Knowns and unknown samples are pre-processed separately in separate areas at separate times to prevent their cross-contamination. And we use dedicated equipment in the laboratory for each step of the process.
May I approach the witness?
Yes, sir.
What I'm showing you is what has been marked for identification purposes as States A. Are you familiar with States A?
Yes.
How are you familiar with it?
I recognize the FDLE case number and my mark and initials on the outside packaging.
And can you describe how this envelope was when you received it for testing?
It was in a sealed condition.
With blue evidence tape? Well, what color evidence tape do you put back on the package when you're done with it?
I put red evidence tape on it.
Right. And does this look to be in substantially the same condition as when you sealed it with red evidence tape when you were done with it?
Yes, it does.
What is A?
It is a DNA card from Nick Presha.
And what is a DNA card?
A DNA card, in this case a bloodstain card, is a piece of filter paper where blood from a particular person is dried down onto it.
And do you do anything to test whether or not that's actually blood, or do you forego that sort of testing on a DNA blood stain card?
We forgo the blood testing on a stain card and take it straight to DNA testing.
What is DNA?
DNA stands for deoxyribonucleic acid. It's your genetic blueprint. It's found in virtually every cell of your body, and within those cells, your DNA is the same. Your DNA profile is determined at conception and remains the same throughout your lifetime. Only identical twins will have the same DNA profile at all 13 or 15 locations that I test for.
How much percentage-wise of DNA would you, me, and anybody else walking around on the planet as a human being share in common?
It would depend on whether or not that individual is related to another. People will share alleles or DNA information at multiple locations, especially if they're related.
And I believe you started to testify about it, but has science determined that there are locations that are different from person to person?
Yes.
And how many different locations, depending on which kits you use, do you test to see the different locations?
In the previous kit that we used, we tested for 13 locations. So for the front end of the testing, in this case, I tested for 13 locations and a gender identification marker. At the back end in the kit that we're using right now, I tested for 15 locations and a gender identification marker.
All right, what is the first step? You open up this envelope that is labeled A, and what do you do to start the DNA analysis?
In the case of a blood stain card like this, I would take a cutting, place that cutting into a tube, and start the extraction process.
And by cutting, you're just clipping a piece of paper, the filter paper?
Yes, I take a sterile scalpel and I take a small cutting from the blood stain card.
How big is that cutting?
It depends on what the blood looks like. If it looks like it's red, fresh, not degraded, then I would take a small cutting forward.
What do you mean by degraded? Does DNA degrade?
It can under certain conditions if it's exposed to the environment. If, say, the blood is left outside for several days or exposed to harsh conditions, Florida's humidity and all of the rain are not good for DNA samples. It tends to break them down over time.
All right, and so you take a cutting and you put it into, did you say a tube?
Yes.
And what do you put in there with the cutting?
I put chemicals, and then I apply heat, and that process will take the DNA and remove it from the cells and from whatever it may be deposited on.
And how do you go about doing this? But you just apply heat to it?
I add the chemicals, and then I put it in the tube inside a heating block, and it maintains the temperature for a certain amount of time called an incubation period.
What do you do after you've incubated and heated this tube with the clipping and the chemicals?
Once I've completed the extraction process, I move on to a quantitation step. That step tells me how much DNA, if any, I have recovered from that extraction process.
And is there any sort of amplification process to increase the number of the parts that you're looking to test?
Yes, that's the next step of the process. That's amplification. I target and copy the DNA at the 13 or 15 specific locations and gender identification marker that I test for. That process allows me to copy the DNA at that location millions and millions of times. So I'm essentially making like Xerox copies of the DNA, and then I take that on to the processing step.
And what is that process of amplification called?
It's called PCR, the polymerase chain reaction.
And that just requires more chemicals, and what else do you do to make this happen?
I add chemicals from the kit that we purchase from the manufacturer. I add that, then put the samples into what's called a thermal cycler. This thermal cycler heats and cools the sample in cycles, and that's how the amplification occurs. It basically replicates the process that your cell undergoes when it replicates DNA within your own body. We're just doing it chemically.
All right, after this process, what's the next step?
The next step is processing. That's where any DNA that I have obtained through the previous processes is collected into instrument software for analysis.
And what are you looking for at each of these locations? What specifically are you looking for so that you can differentiate one person's DNA from another's?
The type of DNA testing I do is called STR testing, short tandem repeat testing. The DNA at the locations that I look at, they're repetitive elements. It's a short sequence of DNA that repeats itself over and over again at these locations. The testing process simply counts how many times those repeats occur, and that's what makes up your DNA profile.
So for example, at one of these locations, I might have a 1 and you might have a 2 or something else?
Something like that, yes. My own DNA profile, I'm a 15-15 at one location. So at one copy of my DNA, the repetitive element repeats 15 times, and on the other copy, it also repeats 15 times. I received a 15 repeat sequence from my mother and a 15 from my father. So my profile at that particular location would be 15-15.
And so some people might, like you, have the same number at the same location, but might others have different numbers?
Yes. Another individual might be a 16-17 at that location, or at another location, they might be a 25-35.
Now, with A for identification, you took it through all these different steps?
Yes, I did.
Were you able to develop a DNA profile?
I was.
And did you use the process that gave you 13 or 15 locations with this particular piece of evidence?
13 locations.
And a gender identification marker?
Yes.
Stop by and see why DeLand Nissan delivers.
Get $4,000 off MSRP on a new Altima or drive it for just $170.
May I approach, Judge?
Yes, sir.
Showing you B for identification now. You recognize B for identification?
I do.
What do you recognize it as?
A DNA card from Jeremy Stewart.
And is that similar in nature for the DNA card we just discussed for State's Exhibit A?
It is.
And by the way, did State's Exhibit A have an FDLE number associated with it?
It did.
And what was A's?
K1.
All right. And now does B for identification have an FDLE number associated with it?
It does.
And what one is that?
K2.
And does the K have any significance?
A K indicates that the sample is a known sample. It came directly from an individual, which means that I could use this sample for comparison purposes to evidence samples in the case.
And when you received B for identification, was it sealed with blue evidence tape?
Yes.
And when you were done testing it, did you seal it with your red FDLE evidence tape?
I did.
And does it appear to be in substantially the same condition as when you last saw it?
Yes.
Did you take B for identification through the same testing process as A for identification?
Yes, I did.
Were you able to develop a DNA profile?
Yes.
Showing you C for identification. Do you recognize C for identification?
I do.
And what do you recognize it as?
Buccal swabs. Represented as being from Angela Patterson.
And when you received this envelope, was it sealed with blue evidence tape?
Yes,
When you were done with it, did you seal it in red evidence tape and put your initials over it?
I did.
And when you open up the contents of C, tell us what a buccal swab looks like.
A buccal swab looks like just a cotton tip swab, sort of like a Q-tip, and it's used to swab the inside of an individual's cheek for DNA.
And was it in a box or anything?
Yes.
And so how do you go about testing a part of the Q-tip or the cotton swab?
I open the item up, I take out the swabs, and I take a small cutting from one of the swabs, place it in a tube, just like with the bloodstain card, and I take that sample forward to DNA testing.
And again, how big of a cutting are you taking, if you can tell us?
In terms of a buccal swab, a very, very small cutting.
And why a very small cutting?
Because if you can imagine, the inside of your mouth contains a lot of DNA. So even swabbing lightly, you're going to pick up a lot of cells on the swab, and the DNA testing process is very, very sensitive. So if I have too much DNA, then I can't tell what the profile is. Sometimes it causes problems with interpretation, so I have to go back and start over. So I want to take the right amount of sample forward so that I hit the right spot in the amplification process to get the best results possible.
Do you have any idea how few cells it's actually required to generate a good amplification?
I don't recall how many cells. In the process, we target 1 nanogram of DNA, which is 1/1,000,000th of a gram.
Okay. And were you able to develop a DNA profile from the cutting of the Q-tip swab from C?
Yes.
And at this time, the state would move C for identification into evidence. It is. It's State's Exhibit 165. All right. Showing you now D for identification. Do you recognize D for identification?
Yes, I do.
What do you recognize D as?
Buccal swabs from Jesse Brandon Davis.
And did you receive this package in a similar state as we have discussed for A, B, and C?
Yes,
And when you were done testing it, did you seal it up as we've discussed with other exhibits as well?
I did.
And what were the contents of D for identification?
Buccal swabs represented as being from Jesse Davis.
So again, long Q-tip inside a little cardboard box, or perhaps
Yes.
2 of them.
Correct.
And were you able to develop a DNA profile from that through your process?
Yes.
Your Honor, at this time, the state will move D for identification into evidence.
Any objection?
Objection relevance as to Mr. Rodriguez.
Overruled.
Showing you E for identification. Are you familiar with E for identification?
I am.
And how are you familiar with it?
I recognize by the FDLE case number and my initials. It's 2 buccal swabs represented as being from Hector Rodriguez.
And is there an FDLE number associated with this item as well?
Yes,
And what is that?
K11.
And did you receive this package in a similar state as we discussed about with the other exhibits?
Yes,
And did you reseal it with red evidence tape with your initials over it when you were done testing?
I did.
And what were the contents of E for identification?
Buccal swabs represented as being from Hector Rodriguez.
Were you able to develop a DNA profile off of E for identification?
Yes, I was.
Your Honor, the state would move E for identification into evidence.
Any objection?
No objection.
Be accepted.
Did you also receive buccal swab standards from somebody named Patricia Davis Hall, FDLE number assigned to it K12?
Yes.
Did you also receive buccal swab standards from somebody said to be James Wilbur Hall, FDLE number K13?
Yes.
Did you also receive buccal swab standards from somebody said to be Jeffrey Vackel, FDLE number K14?
I did.
Did you also receive buccal swabs from somebody said to be Carl Spain, FDLE number K15?
Yes.
Did you receive something labeled as Q12 representing to be 2 swabs from 3 spent casings?
May I consult my report?
Yes.
Did you say Q12?
Yes, ma'am.
Yes.
And Is it represented in your report which casings the swabs were said to come from, FDLE numbers?
U12, DNA swabs from 3 spent casings.
And were you able to develop any profiles from these swabs?
No.
And are you familiar with how a semi-automatic firearm works?
No.
Are you familiar with what effect heat might have on DNA and the degradation of DNA?
Yes.
And what does heat do to DNA?
It breaks it down.
So hypothetically, if a firearm expels a cartridge by an explosive mechanism, oxygenating gunpowder and whatnot, and generates a lot of heat, could that degrade DNA in your opinion?
Yes, the heat and the chemicals could both do that.
Is it unusual in any way for you to not develop DNA profiles off of spent shell casings?
It's not unusual at all.
In fact, how often do you ever develop a DNA profile off a swab said to have come from spent shell casings?
Hardly ever. Maybe never.
Did you also receive something labeled as Q13, represented to be 2 swabs from a lip of a Heineken beer can?
I did.
Were you able to develop DNA profile off of that?
I was.
And did that compare to any known samples of K buccal swabs provided to you?
The DNA profile from Q13 matched the DNA profile from James Danforth.
At all the locations you test for?
At all the locations I test for, yes.
Now, has anybody figured out if there's any statistical significance to matching profiles at different locations?
Yes, I perform a statistical analysis with every match or inclusion that I make.
And what statistical model are you using to do that?
Calculate a probability that I'm going to test one random individual in a population and be able to match that person to the profile that I obtained. It's basically the probability of a coincidental match. And which,
I believe there are two different statistical studies that the FDLE relies upon depending on which kits you use to make statistical calls?
Yes.
Which one is relevant to the Heineken to James Danforth's DNA standard?
Based on when I made the match, I used a validated statistical database put together by Dr. John Butler.
And can you tell the jury a little bit about how Dr. Butler went about doing his study?
Samples were purchased from blood banks and a biotechnology company. They were run using the Identifiler Plus kit.
Is that one of the kits you used?
Yes.
Okay.
That is one of the kits that I used. The samples are put into a database. It's validated, statistically tested, published, and peer-reviewed. The ethnic groups that are tested in that particular database are the Caucasian, African American, and Hispanic populations.
And what are the chances statistically based on that study of finding another person with the same profile as Mr. Danforth as was found on the lip of that Heineken can?
The probability of a coincidental match for 13 loci here is 1 in 24 quadrillion.
So let's talk about a couple of the words you used here. A loci, what does that translate to from Latin or whatever that is?
Loci indicates the locations that I test for on the DNA. One location would be one locus, multiple locations would be loci.
And so you might be 15-15 at a locus, and that would be the proper terminology for that?
Yes.
And so you tested 13 of these different locations?
Yes.
And is there any relationship whatsoever, like if I have a 1-2 at one location, does that have any statistical significance to what I might be at the other locations, or is it independent?
It's independent.
And so is there a word for the multiplication that you do to figure out the chances of all 13 locations?
In terms of calculating the statistic, I use the frequencies in the database and I calculate the frequency at each individual location where I get DNA results. Then I take those frequencies and I multiply them together using the product rule to get an overall estimate as to how often I would expect to see the entire profile across the locations that I have results for.
And so how many zeros is a quadrillion?
Quadrillion
Can you
is,
explain that big number to the jury?
a quadrillion is a one with 15 zeros after it.
And about how many people are on Earth if we're going to compare that to Earth populations?
Approximately 7 billion.
And so we can say that it's a match. His profile, Danforth's, to
The
the...
DNA profiles are the same, yes they
Okay.
are.
Showing you F for identification now. Are you familiar with states F for identification?
Yes.
And how do you recognize states F?
The FDLE case number, my mark, and initials.
And did you receive it in a fashion, in a state that we've talked about with the other exhibits sealed up by blue evidence tape and labeled by the Orange County Sheriff's Office?
Yes.
When you were done testing this item, did you seal it in red evidence tape and put your initials over the tape?
Yes.
And what was inside of F for identification?
2 swabs for DNA from the lip of a Miller High Life beer can,
And
or beer bottle, excuse me.
was an FDLE number assigned to this exhibit, F for identification?
Yes.
What is that?
Q14.
Your Honor, the state would move to publish 30 to the jury.
You may.
What does the package label say that Q14 came from?
The lip of a Miller High Life beer bottle.
Were you able to develop a DNA profile off of this item?
I was.
First, the state would move F for identification into evidence.
Any objection, defense?
No, Judge.
Be accepted. Next number.
At how many locations were you able to develop a profile on State's Exhibit 189, formerly F for identification?
9 loci were eligible for statistical analysis.
And how does that happen? How do we not be able to get all 13 or all 15?
Sometimes there's just not enough information, there's not enough DNA left behind, or the sample could be degraded.
And at these 9 locations, did you compare them to the knowns or the Ks that were submitted as buccal swabs being representative of people involved in the case?
I did, but to be clear, the DNA profile was eligible for statistical analysis at 9 locations, but I use all of the information that I obtain in the profile to make the comparison.
Okay. And so by that, are you able to exclude people having provided DNA to that 9 location sample?
Yes.
And who were you able to not exclude out of the knowns that were provided to you?
Jesse, Brandon Davis, and Hector Rodriguez are included as possible contributors to the mixed DNA profile from Q14.
Let's talk about this word now, a mixture. What do you mean? How do you know there's a mixture there, and what does that mean?
A mixture is when a DNA profile has more than, or 2 or more donors, more than 1 donor.
And how can you tell that?
When I see the final product of the DNA profile, I see that as peaks on a graph. Based on how many peaks are present at each of the locations that I test for, I can determine whether the DNA profile came from 1 person or more than 1 person.
So for example, if it just tested out as having 1 and 2 at that location, that could come from how many people?
That could come from 1 person.
But if there's a 1, a 2, and a 4, now what?
It may be that there's an additional contributor to the sample. I have to make the evaluation based on all of the results that I get.
And based on your opinion and your expertise, what are the statistical chances of somebody besides Jesse Davis having provided the DNA that you found on that sample?
For 9 loci, the combined frequency of occurrence of this profile is 1 in 160,000 Caucasians, 1 in 45,000 African Americans, and 1 in 80,000 Southeastern Hispanics.
And were you able to make a determination as to the statistics as to what the chances are for a random person other than Hector Rodriguez contributing to the sample on the DNA?
In terms of a mixture sample, the statistic is always applied to the Q, to the question sample in the case, so the statistic will always be the same. So the number is the same for Jesse Davis, and it's the same for Hector Rodriguez.
And what was that statistic for Southeastern Hispanics?
For Southeastern Hispanics, 1 in 80,000.
Did you receive an item that ultimately was labeled as Q15 for FDLE purposes?
I did.
And was that said to be swabs from a cartridge casing?
Yes, it was.
Were you able to develop a DNA profile?
No, I was not.
Showing you G for identification. Are you familiar with G for identification?
Yes.
And how are you familiar with G?
I recognize it by the FDLE case number and my mark and initials.
And did you receive this package in a state similar to that we have described already for the other packages?
Yes.
And when you were done with it, did you seal it and put red evidence tape with your initials over it?
I did.
And does it appear to be in substantially the same condition as when you last saw it?
Yes.
Is there an FDLE number associated with G for identification?
Yes.
And what is that?
Q16.
And what is it said to represent?
2 swabs of suspected blood from the door frame.
Your Honor, at this time, the state would move G for identification into evidence.
Any objection?
No, Your Honor.
Be accepted.
State would move to publish 131.
Yes, sir.
Referring back to State's Exhibit 190, which was G for identification, and which is Q16 for your purposes at FDLE, what were the contents of 190?
2 swabs of suspected blood from the door frame.
And what were the contents? Similar to the packaging for the other swabs and the Q-tips in the boxes?
Yes.
Were you able to develop a DNA profile?
Yes, but I first tested these samples for the possible presence of blood.
How do you go about doing that?
In terms of looking for blood, I take a small sampling of the swabs, usually a red-brown stain, and I add a series of chemicals to that sampling. If I see an immediate pink color change, that sample is positive for the possible presence of blood.
And do you leave it at that, the possible presence of blood, or is there further testing?
Normally it's left at that because I'm going to take the sample onto DNA testing. If the sample happens to be some sort of false positive, I would probably not obtain a DNA profile from those types of substances.
After you did that serological testing, did you do the DNA testing?
I did.
Did you develop a profile?
I did.
Was it a mixture or just associated with one person?
It was a complete DNA profile from one individual.
And did that match any of the known profiles that were submitted to you?
Yes, it did.
And whose was that?
It matched the DNA profile from Jesse Brandon Davis.
At all the locations?
Yes.
And what are the statistical significance or the chances of another random person having that same profile that was left in Q16?
The frequency of occurrence is 1 in 10 quadrillion Caucasians, 1 in 460 quadrillion African Americans, and 1 in 19 quadrillion Southeastern Hispanics.
Showing you H for identification. Are you familiar with this item?
Yes.
And how are you familiar with it?
I recognize it by the FDLE case number, my mark, and initials.
And when you received it, was it in a similar state as the previous exhibits that we've discussed?
Yes.
When you were done with it, did you seal it in red evidence tape with your initials over it?
Yes.
And what were the contents of H for identification?
2 swabs from the east living room wall.
And is there an FDLE number associated with this item?
Yes.
And what is that?
Q17.
At this time, the state would move H for identification into evidence.
Any objection, defense?
No objection.
Accepted. Next number, Madam Clerk.
And what were the contents of 191?
2 swabs.
Packaged in a similar fashion?
Yes.
And were you able to develop a DNA profile from this sample?
Yes.
Actually, did you test it for blood first?
I did.
And was it positive for blood?
Yes, it was.
And then were you able to develop a profile?
Yes.
And was this profile reflective of a mixture or of just one person's DNA?
It was a mixture.
And did this mixture have any statistical significance or meaning to any of the known samples provided to you?
Yes.
And which people were not excluded?
Jeremy Stewart and Jesse Brandon Davis are included as possible contributors to this mixed DNA profile.
And what was the statistical significance? The statistical significance would be same for each since it's a mixture, is that correct?
That is correct.
And so what are the chances of somebody out in the random population having contributed to this DNA mixture besides Jeremy Stewart and Jesse Davis?
The combined frequency of occurrence of the mixed DNA profile is 1 in 1.1 million Caucasians, 1 in 4.1 million African Americans, and 1 in 1.5 million Southeastern Hispanics.
And how many locations out of the 13 or 15 that you tested does that reflect?
This is all 13 locations.
And are we still using the first statistical model in the first kits, or have we gone to the second kit and second statistical model yet?
This one is the 13 locus testing, and I use the FBI's database to calculate this.
Okay. And what's your familiarity with the FBI database?
The FBI database is the database we've used for years and years. It's composed of individuals from the Caucasian, African American, and Southeastern Hispanic populations. It's validated, tested, published, and peer-reviewed, and accepted in the scientific community, just like the other database that I use.
Did you also receive an item labeled Q18, gene shorts, for DNA testing?
Yes.
Did you first examine the item to see if there was anything worth testing for the sign of blood?
Yes.
And did you find any signs worth testing for blood?
Yes, I tested several stains for the possible presence of blood, and those stains were positive.
Were you able to develop a DNA profile on any of the stains?
Yes, I developed either a complete DNA profile or a partial DNA profile from all of the stains that I tested.
And did they have any meaning or statistical meaning to any of the known profiles submitted to you?
No, they didn't match anyone.
Q19, representative 2 swabs of the floor. Did you receive that item?
I did.
And did you test that for the presence of blood?
Yes, it was positive for the possible presence of blood.
Were you able to develop a DNA profile?
No, I was not.
Did you receive something labeled Q20, swabs represented as the left rear passenger's door,
Yes,
interior handle? Did you test that for blood?
I did.
What were your results?
I'm sorry, it was negative for the possible presence of blood.
Did you go on to DNA test?
Yes,
Now, why would you do that when it's negative for blood?
because this sample came from a passenger door handle So blood might not be the only source of DNA on that swab. There could also be skin cells left behind by someone who touched the door handle or opened the door to get inside the car.
Now, is that what people call touch DNA?
Yes, it is.
So our skin cells have that nuclear material that has DNA in it too?
Yes, they do.
Did you receive something called Q21?
Yes.
Was that said to be swabs from the right rear passenger door interior handle?
Yes.
Did you test that for blood?
Yes, I did.
Was it positive for signs of blood?
No, it was negative.
Did you go on to DNA testing?
I did.
Were you able to develop any profiles?
I obtained a limited profile, but it was not interpretable.
What does that mean?
It means that I didn't get enough information to be able to say anything about who may have donated to that sample.
Did you receive something labeled Q22?
Yes.
And what was that said to be?
One rolled up rug from the rear seats.
Did you test or did you examine that for signs of blood worth testing?
Yes.
Did you find any?
I found 3 stains that gave chemical indications for the presence of blood.
And did you test those stains for DNA?
Yes.
Now, how do you go about doing that? When you have an item like that that's not already swabbed, do you make swabs yourself?
Yes, I take samples myself.
And how do you do that?
I take a small cotton tip swab, and I moisten it with deionized water, and I take it to the item and I swab the stains. Take sort of a rubbing onto the swab, and then I take that swab onto DNA testing.
And then you take a clipping like you would the other clippings?
Yes.
Okay. And forgive me if I've already asked you, did you develop a DNA profile from Q22?
From the first 2 stains, no, I did not obtain any DNA results, but from the 3rd stain, I obtained a mixed DNA profile.
And did any of the known samples that were provided to you in regards to this case, were there anybody not excluded?
Angela Patterson was included as a possible contributor to that mixed DNA profile.
Anybody else?
No.
Any statistical significance you can attribute to finding somebody else besides Angela Patterson that would have donated to that stain?
For 4 loci, the combined frequency of occurrence is 1 in 210 Caucasians, 1 in 100 African Americans, and 1 in 190 Southeastern Hispanics.
Did you receive something labeled Q23, trunk liner?
Yes,
Did you examine that item?
I did.
And did you see anything that showed signs of being worthy for testing for blood?
Yes, I found 1 stain that gave positive results for the possible presence of blood.
Now, when you're testing for blood, does your test rule out other organisms besides humans, or does it show animal blood as well?
No, it will show animal blood as well. The test tests for hemoglobin, so any organism that has hemoglobin is going to test positive if that blood is present.
Okay. So tested positive for blood. Did you develop a DNA profile from Q23's stain?
No, I did not.
Did you receive something labeled Q24, being swabs from interior right rear door?
I did.
And did you test it for blood?
Yes, it was positive for the possible presence of blood.
Did you develop any DNA profile?
Yes.
And was any of the known samples provided to you in this case not excluded?
No.
Q25, did you receive something called Q25 representing swabs from metal floor inside trunk, right rear corner?
Yes,
And did you test that for blood?
I did.
Did it show indications of blood?
It did.
And did you develop a DNA profile?
I obtained a partial DNA profile.
And was any of the known samples provided to you in this case not excluded?
Everyone was excluded.
Showing you what has come into evidence as states 102. It was formerly I for identification. Are you familiar with this item?
Yes, I am.
How are you familiar with it?
I recognize it by the FDLE case number, my mark and initials.
And did you receive this package in a similar fashion as we've stated for the other packages?
Yes,
And when you left that package, did you seal it with red evidence tape with your initials over it?
I did.
And so now, of course, it's open. Would you agree?
Yes,
Now, is this an item that you examined and tested did you examine the contents of this item to look for possible signs of blood?
I did.
And what were the contents of I for identification 102 in evidence?
one white T-shirt.
And did you find anywhere on the shirt that was deemed worthy of testing for blood?
Yes, I found several areas deemed worthy of testing for blood. One of those areas was positive.
And which area was that?
It was a small stain on the left chest area of the T-shirt.
And were you able to develop a DNA profile from that stain?
Yes.
Did that DNA profile have any statistical significance to any of the known samples provided to you in this case?
DNA profile that I obtained was a partial DNA profile. That partial DNA profile matched the profile from Jeremy Stewart.
At how many locations?
I had results at 8 locations.
And so what are the statistical chances of somebody besides Jeremy Stewart of having provided that contribution to the sample?
The chances of testing any random individual in the population and having them match this profile is 1 in 18 billion Caucasians, 1 in 95 billion African Americans, and 1 in 13 billion Southeastern Hispanics.
And again, what's the population of the Earth?
7 billion, approximately.
Always changing.
Definitely.
Did you receive something labeled Q31 for FDLA purposes, represented to be shoes from Hector Rodriguez?
Yes,
And did you call the left shoe 31A and the right one 31B?
I did.
Did you examine it for any signs of possible blood?
Yes.
Did you find any possible signs of blood?
No, I did not.
Did you do any swabbing for DNA testing?
No, I did not.
No further work on that item.
No further work on that item, no.
Did you receive something called Q32, represented as being genes from Hector Rodriguez?
I did.
Did you examine it for signs of blood?
Yes.
Did you find any possible signs of blood?
No, I did not.
Did you do anything as far as trying to test for DNA?
No, I did not.
Did you receive something labeled Q33, represented as a beaded necklace from Jesse Davis?
I did.
Did you examine it for signs of blood?
Yes.
Did you find any?
No.
Did you attempt to swab it or do anything to develop a DNA profile?
No, I did not.
Q34, represented as being a wristwatch from Jesse Davis. Did you examine that item?
I did.
Did you find any signs of blood?
I did not.
Did you do any DNA testing?
No, I did not.
Did you receive something called Q36 that was represented as being 2 swabs from a 7.65 caliber Manhattan pistol and magazine?
I did.
And straight to DNA testing, or did you test that for signs of blood?
Straight to DNA testing, and I did not obtain a DNA profile.
And is it unusual to not develop DNA profiles from swabs when the swabs are looking for touch DNA?
No, it's not unusual at all.
Does that mean the item was never touched by a human being?
No, it does not.
What are the other possible explanations in your opinion?
It could mean that it wasn't touched long enough. Touch samples are highly dependent on how much contact there is. So if there's not a lot of contact between an individual and a particular item, they may just not leave enough cells behind. Also, different individuals leave different numbers of cells behind when they touch something. So I might touch something and leave a lot of cells behind, and someone else might touch something and not leave hardly any cells behind. There's also what happens to the item in between the time it's handled and the time that the sample is collected. And even the way the sample is collected can have an effect on what kind of profile I might get.
And is it also possible that the cells might have once been there and no longer be there?
Yes,
And does time have any effect on that?
it can, yes.
Heat,
Yes,
wiping it down with
Yes.
even just water.
yes,
What about with chemicals that might be cleaning agents?
yes.
Are cleaning agents good for DNA?
No.
Showing you J for identification. Are you familiar with J for identification?
Yes.
How are you familiar with it?
I recognize it by the FDLE case number, my mark, and initials.
And did you receive it in a state that we've already described for the previous exhibits?
Yes,
When you were done with it, did you seal it in red evidence tape and put your initials over it?
I did.
Was there an FDLE number assigned to this particular item?
Yes.
And what is that?
Q37.
And what were the contents of J for identification?
Swabs for DNA from the ends of tape.
And did you do any testing to see if blood was present?
I did.
Were signs of blood present?
Yes, the swabs were positive for the possible presence of blood.
Your Honor, the state would move J for identification and evidence.
Any objection?
As to relevance, Judge?
Would parties approach for that one, please? Okay, the defense's objection will be sustained at this time. Ladies and gentlemen, we're going to take our post-lunch break, okay? Everyone remember my admonitions, not to discuss this case among yourselves. Please leave your notepads on your chairs. We're going to take about a 10-15 minute break so we can use the facilities, okay? Please retire with the deputies into the jury room. Thank you.
Jury exiting.
Okay, the jury's left the courtroom. We'll take a 10-15 minute break now. Does your client need these facilities? Okay, we'll take care of that now. While they're doing that, will the lawyers approach off the record for a second, please? Thank you.
Stand down?
Well, I'm sorry?
Can the witness stand down?
Sure, she can stand down.
I would like to use the facilities too, thank you.
You're more than welcome to. Yes, ma'am.
Thank you.
State Florida, ready to proceed?
Yes, sir.
Defense?
Yes, sir.
Mr. Rodriguez is present with counsel and the interpreter. Let's bring in our jury, please.
Jury entering.
State of Florida, recognize the presence of the jury.
Yes, sir.
Defense?
Yes, Your Honor.
Okay, Mr. Rodriguez is present with the interpreter as well and counsel. Ms. Crumbly, you're still under oath on the witness stand. State, you may continue your inquiry.
Thank you. Going back to J for identification purposes, what was the contents of J?
2 swabs for DNA from the ends of tape.
And did you test those for the presence of blood? I believe we answered that before, but I'm not sure. It's been a break.
Yes.
And then did you go on to DNA testing?
Yes, the samples were positive for the possible presence of blood, and then I took the sample on to DNA testing.
And was any of the known profiles submitted to you not excluded from contributing to J for identification?
Yes, I obtained a partial DNA profile, and that DNA profile matched Jeremy Stewart.
And to what statistical significance?
For 12 loci or locations, the frequency of the profile is 1 in 190 trillion Caucasians, 1 in 3.4 quadrillion African Americans, and 1 in 290 trillion Southeastern Hispanics.
Trillions is the one that comes before quadrillions?
Yes,
And that's still more than billions.
it is. A trillion is 1 with 12 zeros.
Did you examine something labeled as Q43, a cigarette butt from marker 12 at Cady Way?
I did.
And did you test that for blood or just go on to try and test for DNA?
Just went straight on to DNA.
How do you go about testing a cigarette butt for that?
When I open it up and I look at the cigarette butt, I look at the filter portion, and I take a cutting of that filter portion because that's the part that would have been in the person's mouth as they smoked the cigarette.
And is the heat from the smoke, cigarette smoke, is that good for DNA?
It's not good for it, but when there's a filter, it shields that part of the sample from the extreme heat, and a lot of times I'm still able to get a DNA profile.
Were you able to off of Q43?
No.
And we'll take Q44, Q45, and Q46 together. Were those also represented as being cigarette butts from Cady Way?
Yes.
Were you able to develop a DNA profile off of any of those 3 items?
I was from Q44. For Q43 cigarette butt, Q45 cigarette butt, and Q46 cigarette butt, I obtained no DNA results.
Let's talk about Q44. Did any of the known samples that you were given to develop profiles possibly contribute to the DNA found on Q44?
No.
Q47, did you examine something called Q47, which was represented as 2 swabs of a blue cup on the rear left floorboard?
Yes, I did.
Were you able to develop a DNA profile?
Yes.
And were any of the known samples that were submitted to you in this case possible contributors?
Everyone was excluded with the exception of Mr. Jesse Brandon Davis. I couldn't make a determination as to his presence within the DNA profile that I obtained, which was a mixture.
Okay. Q48, did you examine something called Q48?
I did.
And what was that?
A glove.
What type of glove? Plastic, winter, skiing?
I'd have to check my notes.
Go ahead. And did you, how do you go about trying to develop something to test for DNA from plastic gloves?
When the gloves are presented to me, I don't have any way of knowing which part of the glove is the inside or the outside. So I take a cotton tip swab moistened with water and I swab the apparent outside of the glove. And then I take a separate swab and I swab the apparent inside of the glove to determine if any skin cells were left behind by anyone who had contact with those gloves.
Were you able to develop a DNA profile with either of those two swabs?
Yes.
And was any of the known samples provided to you in this case included as a possible contributor?
I was not able to include anyone, no.
Showing you K for identification. Are you familiar with K for identification?
Yes, I am.
How are you familiar with it?
I recognize the FDLE case number and my initials and my mark.
And when you received that item, was it packaged with blue evidence tape in a similar fashion as to what we've been previously discussing?
It was, yes.
And when you were done with this item, did you seal it in red evidence tape with your initials over it?
Yes, I did.
What was the contents of K for identification?
Cigarette butt on the rear right floorboard.
State would move to publish 159.
May.
State would move K for identification into evidence.
Any objection, defense? Will be accepted. Madam Clerk?
Is there a Q number associated with this exhibit?
Yes, Q49.
And again, what were the contents of 192?
A cigarette butt on rear right floorboard.
And were you able to test it for DNA?
Yes.
Similar fashion as you described before with the other cigarette butt?
Yes.
Did you develop a DNA profile?
Yes, I did.
And were any of the known samples that were provided to you in this case included as a possible contributor to this profile?
Yes.
And who might that be?
The DNA profile obtained from Exhibit Q49 matches the DNA profile from Nicholas Presha.
And to what statistical significance?
For 13 loci, the frequency of occurrence obtained from this DNA profile in unrelated individuals is approximately 1 in 53 quadrillion.
Showing you Y for an identification, which is states 133 in evidence. It's also, I believe, Q50 for FDLE. Is that correct?
Yes.
Are you familiar with this item?
Yes.
How are you familiar with this item?
I recognize it by the FDLE case number, initials, and my mark.
And did you receive that item in a state similar to the other packages that we've had you testifying about?
Yes, I did.
And when you were done with it, did you seal it with red evidence tape and put your initials over it?
Yes, I did.
Did you examine this item for possible signs of blood?
Yes, I did.
Did you find any possible signs of blood?
I found one stain that gave chemical indications for the possible presence of blood.
And did you test that stain for DNA?
Yes, I did.
Did you test any other areas of the item inside of that exhibit? Or just that one?
I tested 2 additional stains that were negative for the possible presence of blood.
Can you repeat your last
Repeat your answer
response?
closer to the microphone.
Sorry. I tested 2 additional stains for the possible presence of blood. Those 2 stains were negative.
Now, these 3 different areas on this item, do you test them together for DNA or separately?
I only tested the positive stain for DNA, but
Okay.
normally it would be separately.
And did you develop a DNA profile?
Yes, I did.
And were any of the known samples that were provided to you in this case included as a possible contributor?
That DNA profile matched the DNA profile from Angela Patterson.
At all the locations you tested?
Yes.
Was that 13 or 15 in that instance?
The profile from the jacket had 15 locations tested, but the standard from Ms. Patterson was a 13 locus test, so I made the comparison at 13, so the statistic is on the 13.
And we'll just say it exceeds the Earth's population by a lot?
Yes, it's 1 in 3.4 quadrillion.
Did you test something labeled Q51, a Springfield magazine?
Yes, I did.
And by magazine, do we mean a paper magazine, or is this like a gun clip?
Gun clip.
And how do you go about trying to get a DNA profile off of a gun clip?
In the similar fashion, as I described before, I take a cotton tip swab moistened with deionized water, and I run the swab over that item in order to try to pick up any skin cells.
And were you able to develop a DNA profile?
I was not.
Did you receive something labeled as a magazine Q52?
I did.
And again, that's a clip for a semi-automatic handgun, not a periodical publication?
Yes.
Were you able to develop a DNA profile?
I was not.
Did you receive something labeled Q53, swabs from a .40 Glock 27?
Yes.
And that was from the pistol, the magazine, and the rounds?
Yes.
Were you able to develop a DNA profile?
Yes, I was.
Any of the people that submitted known samples in this case included as possible contributors to the DNA profile?
No.
Does that mean that none of the people that provided known samples in this case ever handled that item or the items?
It does not.
Q54, submission represented as being from the left fingernails of Jeremy Stewart. Is that something you examined?
Yes.
And did you test it for the presence of blood?
I'm sorry, for Q54?
Yes, ma'am.
No, I did not.
Did you do DNA testing?
I did.
Did you develop a profile foreign to Jeremy Stewart in any way?
I did not.
Q55 represented as being ankle swabs from Jeremy Stewart, A being right, 55B being left. Did you develop a DNA profile in any way foreign to Jeremy Stewart?
I did not.
56, Q56 being represented as ankle swabs from Nick Presha, A being the right ankle, 56B being the left ankle. Did you in any way develop a DNA profile foreign to Nick Presha?
No, I did not.
Q57 represented as being a pair of jeans from Jesse Davis being booked. Did you test any part of that item for DNA?
I tested 2 stains for DNA.
Did you test them for blood first?
I did.
Were either of the stains positive for blood?
There were 2 stains that were positive for the possible presence of blood and 1 stain that was negative. I took the 2 positive stains onto DNA separately.
Did you develop a profile?
For the first stain, I did not obtain any DNA results for that sample. For the second stain, I obtained a mixture.
And were any of the known samples provided to you in this case possible contributors to this mixture sample?
No, the mixture sample was not interpretable.
And is laundry detergent good for DNA?
No, it's not.
Is hot water good for DNA?
No.
Q58 swabs of suspected blood from a register. Did you examine something called Q58 ultimately?
Yes, I did.
And did you test it for the presence of blood?
Yes.
What were your results?
Those swabs were negative for blood.
Did you go on to DNA testing based on that?
No.
I don't have any other questions of this witness.
Cross? When you begin your testing, are you advised where the items were found within the crime scene?
Sometimes, yes.
In this case, were you?
Yes.
Is that by the chart or the map that the state put up on the overhead?
No, I received the items of evidence in the laboratory with a exhibit number, like the K or the Q number, and a description. I don't get any diagrams or anything from the agency.
Now, the DNA for Mr. Danforth that you found on the Heineken beer can, is that the sole DNA that you found or the sole match that you found?
Yes.
Now, I know you don't use the word match, correct?
For a single source profile, yes, I do.
All right. What about when you've got mixtures?
A mixture, it's an inclusion.
And I think you said your lab standard is 13 locations or loci?
It was 13, now it's 15.
Now it's 15?
Yes.
And when did it switch over?
In July, I believe, of 2012.
Okay. So these items were done under the old Rule 13?
Yes.
And with regard to the Miller High Life can Q14, I believe you said there were 9 locations or loci? Is that correct?
Let me verify. There were 9 locations eligible for statistical analysis, yes.
And so that's even below your lab standard at that time, correct?
I wouldn't call it below a lab standard. We run 13 loci as a standard, but I can match 2 DNA profiles at one location, at one marker.
So what does the 9 mean on this then?
The 9 means that I had 9 results at 9 loci that were eligible for statistics. I actually had results at 13 locations, but those results did not meet the criteria to be used for a statistical analysis.
Does that mean they weren't sufficient samples?
The eligibility for statistical use in terms of what the results are at a marker, as I said before, I see the DNA as peaks on a graph. If those peaks don't reach a certain threshold, they're not eligible to be used for statistics. So those peaks just weren't high enough. They were limited DNA samples at those locations.
And on Q14, I believe you indicated that it was a mixture of DNA of Jesse Davis and Hector Rodriguez. Is that correct?
It was a mixture, and those 2 individuals are included, yes.
They're included.
Yes.
And explain to me or explain to the jury what a mixture, inclusive or exclusive is.
In a mixture, I'm looking at the DNA of more than one individual. So there are alleles or DNA information from both of those individuals, or it could be 3 individuals within that mixture. What I do in the comparison is I take the DNA profile from an individual's standard, for example, Mr. Rodriguez or Mr. Davis, and I look to that mixture to see if those alleles or that DNA information is present in the mixture. If that information is present in the mixture, then they are included, and it's possible that they donated DNA to that particular sample.
Now, in this particular item, Q14, I believe you indicated that the statistical analysis was 1 in 80,000 of Southeastern Hispanics. Is that correct?
Yes.
What is the significance of that number as opposed to the quadrillions you were talking about earlier?
Basically, it means that if I tested 80,000 Southeastern Hispanic individuals, I would expect to be able to include 1 individual as a possible contributor to the mixture.
So really that's a very lower standard, isn't it, than the quadrillions? And
yes, it is. It's a lot lower number.
Now, with regard to Q30, and specifically Q30A, the small swab you took out of Q30, did you say that had a partial match to Jeremy Stewart?
Yes, the DNA profile was a partial profile. I obtained a DNA profile at 8 locations.
And what was the other partial profile, or was there one, besides Mr. Stewart?
There was
was there anything else?
No, there wasn't anything else.
Okay. And could you tell us what your statistical analysis was of that?
Excuse me. For Q30A?
30A.
30A. The statistical significance of that match at those 8 locations is 1 in 18 billion Caucasians, 1 in 95 billion African Americans, and 1 in 13 billion Southeastern Hispanics.
And you don't specifically know where these items were found within the crime scene or the ambient area around the crime scene, do you?
No.
And that has no significance in your identification or your analysis, correct?
It does not.
Have a moment, Judge.
Yes, sir. Redirect.
If my known profile is compared to an unknown sample from a crime scene, and it matches at 12 out of 13 locations, but at that 13th location, I'm excluded as a possible contributor, do you call that an exclusion, even though the other 12 matched?
Absolutely. If you don't match at one location, you're excluded. The statistic never comes into play. I don't match people just at the locations where I see their DNA profile is the same. I look at the entire profile or the entire mixture, and I make a determination as to whether or not the profiles match or there's an inclusion, and then the statistical analysis takes place. I'm trying to exclude someone. It is only when I cannot exclude them that they are a match or they are included, and I perform a statistical analysis.
Thank you.
Is she free to go, State?
Yes, sir.
Defense? Yes, sir. Have a good day. Thank you.
Thank you.
State, who's your next witness?
State will call James Danforth.
Good afternoon, sir.
Good afternoon.
You're soft-spoken. Please speak directly into the microphone. Tell us your full name, spell your first and last name.
James Patrick Danforth, JamesDanforth.
Your witness.
Mr. Danforth, where were you living back in April of 2012?
I was living with my girlfriend, with her ex-husband. I know it sounds crazy. And kind of like floating back and forth from there to the mechanic shop. That's where I was trained.
Are you familiar with the area on the Sunday morning, the 15th day of April 2012, where the two individuals were found burning on the Cady Way Trail?
Yes, I'm familiar with that area.
How are you familiar with that area?
It was like a transient drinking spot. So everybody used to hang out and drink there.
Did you hang out regularly at that particular spot and drink back in April of 2012?
Yes.
Do you recall the specific night, the Saturday night and Sunday morning when this happened? Do you recall that time period?
Yes, I do.
Were you at the trail on the night of the 14th or the early morning hours of the 15th, Saturday, Sunday?
No, sir.
How do you know that?
I had a coworker that I did a side job for him at his house that Saturday, and there was just key things that happened that, obviously being questioned in this case, that reminded me,
Do you remember where you stayed on the actual night of the 14th into the 15th?
Yes, I was staying with my girlfriend at the house.
Where was she?
It was at Alafaya Woods. Up by UCF area, Oviedo.
Do you remember prior to that Saturday night, the last time you would have been actually on the Cady Way Trail?
Yes, probably maybe 2-3 days before that.
And that's before the 15th?
Before the 15th.
Do you remember who you were at the trail with the last time you were at the trail prior to this?
I believe I was hanging out with a Carl Spain. Span.
And again, that's prior to the bodies being found, is what I was
Prior to,
talking about.
yes.
You were up on the trail with Carl Spain.
Yes.
What was your favorite beer at the time that you would regularly drink?
Heineken.
You recall at the time you were up at the trail with Mr. Spain that you had consumed a Heineken up there with him?
Yes.
I don't have anything further to this witness.
Cross.
Good afternoon, sir.
Good afternoon.
So you weren't there the early morning hours of the 15th when these two individuals were killed and burnt to death?
No, sir.
Okay. And that beer bottle is not indicative of that?
No, sir.
Okay. But your DNA was found at the scene. You weren't there.
Weren't there.
Now, you said that it was probably 2 to 3 days beforehand that you were there?
Yes.
Okay. How familiar are you with that trail? How often would you go to that area and drink?
Very familiar. Very
Would you frequent in on a regular occasion?
frequent.
Safe to say that, I guess there wasn't that great of a pickup crew or a cleanup crew that traveled in that area?
Could say that.
Okay. So beer bottles and things would be left there for days?
Yes, sir.
Okay. You also said that it was also a frequent hangout place for the transient community.
Mm-hmm.
So any given hour of the day, you could go up there and probably find someone hanging out?
Pretty much.
Okay. Lots of witnesses around?
Maybe.
Okay. People that would see what you were doing?
Mm-hmm. Mm-hmm.
Is it safe to say that, or actually let me ask you this, do you know Hector Rodriguez?
Yes.
Have you ever seen him up drinking in that area?
Yes.
Okay. Would you or the others as you witnessed, would anyone make a conscious effort to make sure that all their cans or bottles were going into the recycling bin or the trash can?
No.
Okay.
I believe we all probably contributed to...
To the litter?
Yeah, to the litter.
Okay. And that litter would stay there for any number of days?
However long.
Okay. So the fact that a bottle was found with your DNA or anyone else's DNA,
Is explainable.
no telling how long that would have sat there, right?
Right.
Okay. Nothing further, Your Honor.
Thank you. Redirect.
One question. You had never seen Jesse Davis up at the trail, correct?
Not on the trail, no.
Nothing further.
Is he free to go, State?
He's free to go, he's released.
Free to go, defense?
Yes, Your Honor.
Have a good day, sir. Thank you. State next witness.
State is going to call Christopher Alfonso. Christopher Alfonso.
Good afternoon, sir.
Good afternoon.
Please speak into the microphone. Please tell us your full name, spell your first and last name.
My full name is Christopher Alfonso, spelled CHRISTOPHER
Your witness, State.
Mr. Alfonso, where were you living back in April 2012?
At 2898 Willow Bay Terrace in Castleberry, Florida.
Do you remember a time in April of 2012 where your vehicle was the victim of a burglary?
Yes, I believe it was the evening, or actually it was the early morning of April 7th, 2012.
where were you where your vehicle was burglarized?
I was staying with a friend on, I believe it was on Shoals Drive in Winter Park.
What, if anything, do you recall that was burglarized from that vehicle?
I had a firearm and a tactical flashlight stolen from the car.
What specific type of firearm was it?
It was a Glock 40 cal semi-automatic.
I'm going to show you what's already been evidence, has already been entered into evidence as State's Exhibit 136. If I can approach the witness.
You may.
Do you recognize that?
Can I pick it up?
We have the gloves on.
Right in front of you.
Yes, I recognize it.
Familiar with the serial number also?
Yes, it's mine.
That was your gun?
Yes, sir. It was registered to me.
Nothing further to this witness.
Cross-examination.
Mr. Alfonso, you described that your vehicle was burglarized.
Yes, sir.
Okay. Is that meaning that whoever took that gun did not have your permission to have that weapon?
No, absolutely not.
Didn't have any ownership interest in that gun?
No.
You didn't sell it to him or anything?
No, absolutely not.
Nothing further, Your Honor.
Redirect.
No, Your Honor.
Is he free to go? Defense.
Yes, Your Honor.
Have a good day, sir. Thank you.
Thank you.
Next witness, State.
autopsy or as part of that?
Yes.
Was that done in this particular case?
Yes, it's done in every case.
I'm going to show you what has been marked as State's Exhibit JAIZIYIXIWIV IU IT IS IR IQ IP IO IN IM IL IK IJ II IH IG IF. If I can approach, Your Honor.
Yes, sir.
Look at these to yourself, sir. Recognize those photographs?
I do.
Are those photographs either taken by your crime scene investigator at the scene or taken during the course of the autopsy, and do they fairly and accurately depict what you utilized in making conclusions in your autopsy and coming to a conclusion as to the cause and manner of death?
They are, and they do.
Your Honor, at this time, I'd like to introduce previously listed items for identification into evidence.
Any objection, defense? To be accepted?
getting back to your your autopsy of Nicholas Presha, I'd asked you about the size of Mr. Presha at the time he was autopsied, and could you report to members of the jury what your findings were?
155 pounds and 68 inches in length.
You also have an opportunity to do an autopsy on an individual by the name of Jeremy Stewart.
I did.
When was that autopsy conducted?
Same day, April the 16th, 2012.
And regarding your findings regarding his particular height and weight, what were your findings?
160 pounds and 70 inches in length.
Showing you State's Exhibit 214. What, if anything, are you looking for when you look at crime scene? Can you see?
1 second. Go ahead.
All right. What, if anything, are you looking for that may assist you in making a determination as to the cause and manner of death when you look at pictures that the investigator takes over the crime scene?
Well, as far as determining the cause and manner of death, the cause of death, it depends on the case. You really don't know at the time that you're looking at it what might be important and what might not be.
Do you attempt to obtain information also from the detectives regarding any information that may assist you in, as part of your autopsy in making a determination as to what may have occurred?
Yes. Well, of course, before I am notified, somebody has already made a determination that an individual has died. So someone is always involved before our office is involved. And with that notification will come background information, apparent injury, position of the body, where the body was found. And usually there is some preliminary investigative narrative, which is what we call it, that attends that case. In this case, I don't recall the specifics of it, but there were 2 bodies found burning off of a bike trail in a public area.
What does the fact that the bodies were found burned, could you tell by doing these particular autopsies how long the bodies were burning prior to the death of either individual?
Well, I couldn't say for certain how long, but I don't think it wasn't too long because the bodies were not nearly consumed by the fire. There were relatively superficial injuries, and there were more extreme injuries to the extremities, but the internal organs were intact. And as a matter of fact, beneath the clothing, there was actually fresh or relatively fresh blood that was present in a semi-liquid and in partially liquid state. So that it could not have been too intense of a fire for too long.
What, if any, significance did you give to the fact that the bodies are positioned in this particular manner?
Well, on the face of it, obviously these are 2 individuals who did not come to this end, most likely by their own devices. It appears that they have been placed there, but in general, it's difficult to make too many assumptions just based on the viewing of the bodies at the scene without more information.
What, if any, significance is there to the fact that the left arm of each individual appears to be in the same position behind the back?
Well, that could be the result of
Sustain, you may re-ask the question or rephrase the question or ask a different question.
Does that suggest anything specific? Is that a normal place where you would expect, if somebody was shot and fell and wound up in that manner, where their arm would be, based on your training experience?
Overruled, I mean, it's sustained, but if he has a basis for that response, he has to establish the basis first.
I don't believe it's a typical pose, but it could be the result of a person falling that way, or a person could have been placed in that position after they died. There's nothing that I can see from that scene picture that would rule that out.
Based on your training experience, are you familiar with, and I want to show you the other photo here. This is State's Exhibit 213. And we're still talking about the position of the bodies, the fact that they appear to be similar from this particular perspective as well. Is that correct?
That's correct.
State, before we go forward, do you want to publish to the jury or just to the witness?
They're already in evidence.
So do you want,
I expect that
they're not being published to the jury?
They are not. All right, I do prefer that they would be then.
Okay.
And then we can go back and show the one. All right, just so we can clear the record, this is State's Exhibit 214. We were talking about the situation with the arm behind the back, the left arm of each. Is that correct?
That's correct.
And then we were talking about 213, where we have the right arm in a similar position of each of them.
Yes. What we see here are two bodies that have been subjected to fire, heat, thermal radiation. And typically, when that occurs in a human body, they will adopt what's termed a pugilistic pose. It's often likened to the pose of a boxer, with the arms flexed at the elbow and the hands flexed at the wrists, and the fingers of the hands also flexed. This occurs because the heat actually cooks the muscle in the body, and it causes that muscle to contract. Now, the reason that it produces flexion is that the muscles in our extremities that cause our arms and fingers to flex are more developed or stronger than the muscles that cause us to extend our arm or our fingers. So the net effect is when a body is subjected to heat after death, that they will flex their extremities and adopt what's termed that pugilistic pose. In this photo, you can see a deceased person there with the arm bent at the elbow. The hand is actually flexed at the wrist, and that's characteristic of bodies that are involved.
So do you have an opinion then as to the positions of the arms regarding when the fire was started versus the final resting position?
Well, it's impossible for me to say that the arms were in a different position, but I can say that if they were in a different position, that the heat would tend to cause the arms to flex. And it's a very strong force. It will eventually, and as it did in this case, actually cause fracturing of the bones.
Now, why wouldn't both hands be in that particular position or depicted in that particular position that you described?
Well, it could be because in this case, they're laying on their left side, and the left arm cannot assume that posture. It would have to pass under the body and toward the front of the body for that to occur.
What can be said by the fact that the left arm in each of these individuals is not in the front of the body and actually behind the body?
It indicates to me that when they died, or at least at some point after their death, before the body heated up, that their left arm, as they were laying on their left side, was behind their body.
Or is there anything that you found in the course of your autopsy inconsistent with their hands being tied behind their back?
There's nothing inconsistent with that.
This is State's Exhibit 212. What did you notice regarding the face of Mr. Presha when he arrived? And this is 211. What did you notice about his face?
Well, his head and his face are almost completely covered by a fabric, cloth, and it has been partially burned. In some cases, thoroughly burned in some areas.
Can you say anything about how tightly that would have been wrapped?
Well, it appears to have been tightly wrapped because it has completely conformed to the shape of the head. There's going to be some shrinkage due to the fire itself, but it appears that it completely conformed to the shape of the head.
What particular areas of the head did it cover?
It covered the entire back of the head and most of the face. If I can refer to my notes here. The upper lip was covered, but not the lower lip, and you could see the tongue protruding. The rest of the head was covered. The rest of the face was covered.
Would that mean that the eyes were covered as well?
The eyes were covered.
What fire damage, if any, did this particular item sustain? I mean, could you tell if there was additional materials that may have burned where you can't exactly come up with a area, or was it pretty much intact?
Well, there was on the outer layer, there appeared to be remnants of boxer shorts because it looked like the waistband of the boxer shorts with the Fruit of the Loom label. That's the only way that I could tell that they were probably boxer shorts. The fabric itself looked like a sweatshirt type material, a common cotton sweatshirt type material, and there was actually one area which looked like the channel in which the drawstring of a hooded sweatshirt would be contained.
You had mentioned something about boxers.
That's correct.
Where was there some artifact of a boxer, or that you believe was a boxer? Where specifically was that?
That was on the outer layer of the head.
Of the head?
Yes.
Based on your examination of the head, how many different types of fabrics or different items did you notice actually on the head?
I think it was two. I think it was the boxer shorts and the sweatshirt material.
Do you believe that the boxer shorts would have been over or on the outside of the sweatshirt material, or vice versa?
On the outside. The boxer shorts were on the outside.
This is States 210. What are members of the jury looking at here?
You're looking at the back side of the body. To the right is the buttocks and the legs, and you can see the right arm and elbow. And there's a fragment of clothing that's still adherent to the body. It's partially burned. It's been partially consumed, but there are still parts of it that remain. And it appears to be blue jean type fabric. It looks like the waistband of blue jeans.
The band that you're talking about in the middle of the picture would be something that would be part of blue jeans?
Looks like it, yes.
Could you tell if he was wearing anything that would have been or constituted a shirt at the time?
We had recovered a white fabric that was present on part of the torso. It looked like it could have been a shirt.
Could you tell if that was tied or on his person or something that was wrapped? I'm going to show you State's Exhibit 209. What does that show there? What item does that show? Can you turn around?
It's part of the fabric. I can't tell from the photo what part of the body that's on. That looks like more of the.
Is that actual jean fabric?
That looks like more of the waistband of the blue jeans. Yes.
So did you have an opportunity in the course of the autopsy to then remove the face gear or what was covering the face of Mr. Presha?
Yes, I did.
Showing you what's been entered in State's Exhibit 206. Tell members of the jury what they're seeing there.
We're looking at the back of the head. You can see the ears. The left ear is dark, partially because it's been burned, but also because there's a shadow there. And then right in the middle of the back of the head, at the level of the mid portion of the ears, you can see a small oval defect, and that is a gunshot wound.
Can you just, can you actually make a marking or an arrow of where that particular gunshot wound is that you see depicted in that picture?
Attempting to draw a circle around it. Right there.
Now, that's one particular gunshot wound. Did you detect any others on Mr. Presha?
Yes, there were two other gunshot wounds. One passed through his head, producing an entrance and an exit defect. And another passed into his head but did not exit.
So this is State's Exhibit 205 I'm showing you?
Yeah. So, that's right. I've underscored this wound. Well, I've actually made it too big. Anyway. I've roughly circled the wound that's closer to his ear. This is the fatal wound. In this case, the bullet passed through the face, upwards through the base of the skull, through the brain, and exited the skull over here on the left side of the head, but did not break through the scalp. And I recovered the bullet from the scalp on the left side of the head. The other wound that's closer to the eye actually passed right through his face and exited on the other side, but did not enter the cranial vault, did not come in contact with the brain.
That's the one with the exit?
That's correct.
Which would have been the fatal, which of any?
The circled wound, the wound that's closest to his ear, would certainly have been fatal. The other wound is potentially fatal and may have been incapacitating, but also could be survived easily.
What attempts, were there any other entrance wounds in Mr. Presha that you noticed during the course of your autopsy?
No.
So is it fair assessment that there were two wounds, two entrance wounds on the right side of his face, one depicted in the back of the head here?
That's correct.
What can you tell us regarding the proximity of the shooter to the entrance wound?
Well, in this case, it's difficult to say anything about that. I mean, the entrance parts of the head, the entrance wounds were covered by fabric. The body was burned. Normally, if you would shoot a person through a fabric at close range, I would see some evidence of soot, blackening of the fibers, and possibly some of that would be transferred onto the skin. But in this case, that fabric is burned and everything is black. And although we looked carefully, we could not see any evidence of close range firing. It doesn't mean that it wasn't close range. It doesn't mean that the muzzle of the gun could not have been actually up against that fabric. I simply can't say that it was.
So you're not able to give an estimate at all based on the fire and the fact that the face was covered with that fabric as to whether this was a contact wound or a wound of longer range?
That's correct.
And that goes for any of the three entrance wounds?
That's correct.
Now, you had talked about, I believe it was gunshot wound 2 having an exit wound. I'm showing you State's Exhibit 207. What does that show?
Can we clear that? Yeah. We see the area above that I've highlighted is an oval defect right there at the hairline in the left temple, and that's the exit wound. That's the exit wound from the bullet that entered closer to the eye and passed through the face and exited, but didn't enter the cranial vault where the brain sits.
What can you tell us about the path that these bullets would have traveled from these gunshot wounds?
Well, the bullets that enter the right side of the face, they all travel, of course, right to left. They both travel toward the rear, and they both travel upward.
They both traveled, I'm sorry, did you say upward?
Upward.
What particular were you describing upward? Are we talking about steeply upward? What specific degree
Well, in
of
the
upward?
case of the bullet that did not exit, it is only a matter of an inch or so. In the bullet that did exit, it's only a matter of an inch or so. In the bullet that did not exit, the one that perforated the brain, it's about 3 plus a little over 3 inches rise from one side of the head to the other.
Did you actually compile a diagram which shows the relative angles of the trajectory of the bullets?
I believe I did a rough diagram. I don't know if it shows the trajectory. It doesn't contain the location of the projectile that was found in the scalp, but it is measured.
Your Honor, at this time, can I approach with State's Exhibit HR?
You may.
This is a diagram that's prepared, termed an injury clarification diagram, which is meant to be a simple graphic representation of the injury.
Does that show the actual locations of the 3 gunshot wounds and also the exit wound on Mr. Presha?
It does.
And would the jury ever be able to appreciate the trajectory of the bullets if you were able to show them by use of this head exactly what you mean by the angle?
Yes, I believe so.
Your Honor, I'd like to exhibit HR as evidence.
Any objection? Be accepted.
Okay, let's talk about gunshot wound 1. Which one are we referring to that you...
That is the one that on the right side of the face that is closer to the ear.
Okay, can you demonstrate to members of the jury, was that one, just to refresh us, does that one did not have an exit, is that correct?
It does not exit, that is correct. It does perforate the skull, so it exits the skull. And I find it roughly on the left side, roughly in this position. Whoop, sorry.
Roughly in that position there?
I'm attempting to, roughly in that position.
All right, what would that suggest to you about the angle that that was traveling? If you could do an example, possibly from the point of the gunshot entrance?
It's about this. It's about like that. Entering here, so it enters here, and it breaks through the skull here, and it's stopped by the scalp. It doesn't exit the scalp. The bullet comes to rest there.
Okay, let's talk about the fatal gunshot wound 2.
That is the fatal gunshot wound, the wound that we just described. That is the one that perforates the brain. It does not exit.
That would be the one closest to the eye?
The one that we just discussed, gunshot wound number 1, is closest to the ear.
So the fatal gunshot wound would be the one closest to the eye?
No, I'm sorry. The fatal gunshot wound is the gunshot wound that we just discussed that is closest to the ear, that passes through the face, into the skull, through the brain, and exits the skull but not the scalp on the left side of the head. So
that would be the fatal gunshot wound?
That is the fatal gunshot wound.
So the second gunshot wound then, which would not be the fatal one, is the one closest to the eye?
Yes. And that one enters about here, and it exits about here. So there's not as much of an angle upward on that one.
But you're still talking about an upward angle?
It's still going upward.
Now, gunshot wound number 3 would be the one to the back of the head, correct?
That's correct.
And tell the members of the jury the path of that projectile.
Well, that bullet goes straight into the back of the head, and it actually doesn't enter the skull. It strikes the skull and moves toward the right, and ends up. If you feel behind your ear, there's a very large solid bone back there called the mastoid, and it actually sticks out a little bit, makes a little point, and that bullet was on the inside of that bone. And that bullet is going pretty much straight in. It doesn't go up or down very much, but it goes to the right. So it's going from the back to the front and toward the right, and it stays fairly level.
What can you tell us about which of the gunshot wounds may have been the first one received by the victim? Is there any way for you to tell?
No.
Other than upward movement, could you tell the path regarding whether it moved left to right, right to left, the projectile? Could you tell the path of the movement of the projectile in these gunshot wounds?
In which one?
In any of them.
Oh, yes, the path is traced.
What was the path of gunshot wound 1, which again is going to be the fatal gunshot wound closest to the right ear?
Well, that bullet penetrates the soft tissue of the face. It causes a fracture of a facial bone, and then it passes into the skull, sort of behind the left eye, then punctures the brain, and then punctures the left side of the skull here, and then is stopped by the scalp.
Can you tell the position of the shooter regarding the side of the body that the shooter would have been, or whether the shooter was facing the body of Mr. Presha when he fired those shots? What can you tell us about the position?
Well, I can't say much about where the shooter was facing, but the muzzle of the gun is pointed to the right side of the face. So if this is the barrel of the gun, the face is here. His face is like that.
Facing to the right side where the gun would be.
That's correct.
And then, of course, in the one in the center, is there anything that suggests that the shooter wouldn't have been behind him at that particular point unless he was reaching out in some manner?
Yeah, again, the gun has to be pointed at the back of the head. However, you can arrange that, whatever contortion either the victim or the shooter can place themselves in. The muzzle of that gun has to be pointed directly at the back of the head.
Can you tell if the victims are upright during any of the shots in the sequence at all, based on the bodies, the position that the bodies were found, and also the position of the trajectory of the bullets?
Well, I can't say that they were necessarily upright, but I can say that it would have been difficult to inflict the wound to the back of the head if they were in the position that they were found. And that would require the shooter to lower himself to the ground and bring the gun down almost to ground level. It's not impossible.
What, if any, of the totality of evidence in this case when there's a fire, what, if anything, do you look for to make a determination if these individuals would have been set on fire prior to the shooting or subsequent to the shooting?
What we look for in a fire death is evidence that the person was alive at the time of the fire. And there are two general ways to do that. One, you can look for soot or products of combustion. Most fires are somewhat dirty, smoky, and if you're alive, you're going to inhale, whether you're conscious or not. You're going to breathe in, and that soot, those particles that make up smoke, will become embedded in the mucus that lines your respiratory passages. The other thing that we frequently look for in fire deaths is carbon monoxide. There may or may not be carbon monoxide in fires that occur such as this, but in this case, in both of the individuals, there was no evidence of soot and there was no carbon monoxide.
Showing you State's Exhibit 203. Asking you what, if anything, is shown in 203.
This is a difficult photograph to orient yourself to. The individual here is laying face up on the autopsy table. You are looking at the left side of the head. Let's see if I can.
What, if anything, can you tell about the amount of blood?
Well, there's a moderate amount of blood in the scalp. but I think that this picture doesn't. At the dot here is the left ear. Now, when an autopsy is performed, in order to gain access to the cranial vault, an incision is made from behind one ear, completely over the top of the head, to a point behind the other ear. So we've got an incision. The scalp is cut. It's then pulled forward and pulled backward, exposing the skull. And that's what you can see here. So the scalp here in this position has been pulled forward, and you're looking at the inner aspect of the scalp now. So this is the face up here. This blood that you see here is the result of this, which is the exit of the bullet. So that bullet passed through the skull here. It got trapped in the scalp over at where you see that blood, and that's the cause of that blood or that hemorrhage that's in the scalp.
Based on the 3 gunshot wounds and, of course, the fatal gunshot wound that you had described entering the right cheek, what damage would have Mr. Presha sustained, and what would have been the effects on him at that time? And I guess that's a complicated way of asking how much movement could have you expected out of him if these shots were shot into him at around the same time?
Well, for the bullet that strikes the back of his head and doesn't enter the brain, doesn't really produce any much in the way of visible injury, I can't say. A person could survive that wound, could possibly be rendered unconscious just because of the area that the bullet struck is very close to the cervical spine, and there could be some shock there, which could cause loss of consciousness. However, I can't determine that. The second bullet, the bullet that struck the right side of his face and exited on the left side, again, it's not an obviously fatal injury. It wouldn't have been immediately fatal. I could see that it could possibly have produced unconsciousness, but also possibly not. The fatal wound, however, the bullet that passes from the right side of the face through the base of the skull and through the brain would have been incapacitating and would have caused him to no longer be able to move or be conscious. Now, there can still be some movement, especially in the immediate aftermath of a gunshot wound to the head. There can still be some involuntary movement or spastic type movement, but I think that there would be no purposeful movement.
If the first gunshot wound, hypothetically, was the gunshot wound that was to the back of the head, is there, based on your examination of that wound and the caliber and what you know about that, can you say for sure whether that would have knocked him completely off his feet and knocked him over at that point if he was upright?
I cannot.
What, if any, other injuries did you notice during the course of your autopsy of Mr. Presha?
I didn't see any other injuries other than those things that would have been associated with the bullets passing through his head. However, there were many areas of the body that were charred, and superficial injuries such as abrasions, like a skinned knee, things like that, superficial cuts, those could have been obscured. Some bruising, superficial bruising, especially in the areas of the extremities that were largely consumed by the fire, those could all have been obscured and may have been present and not detected. I did find on his right calf, there was a small fragment of metal, brownish, dull metal that was embedded superficially in the calf. It didn't produce any significant injury, but it was there. It produced a little defect, and I believe now after investigation of the scene and discussing with investigators that that was actually the casing of a bullet, a cartridge. Not the projectile itself, not the part that's fired, but the part that holds the powder and the part that remains in the gun or is ejected from the gun after it's fired. And that bullet, that cartridge had...
And Jeremy Stewart's father, right behind her in the blue shirt, listened as all of the victims' families have since court began. Today, Jeremy Stewart's mother took the stand.
When was the last time you saw your son?
I saw him on April 14th.
Jeremy Stewart and Nick Presha were shot to death and their bodies burned near the wooden bridge at the Cady Way Trail last April. The